Tonight, on another episode of Fake News Uncovered, we dig deep to bring you the exclusive story of Cloning Project 6449.
In 2016, during the Fees Must Fall campaign, many researchers were prevented from accessing their labs and research came to a halt. Moving science quickly became popular in scientific circles because it developed many innovations that helped to facilitate research in difficult times. On the Moving Science website, we discovered that many of the products are sold out due to the COVID-19 pandemic, as researchers rushed to purchase tools that would keep them safe and at home while still producing results in the lab. One of the most popular services provided by Moving Science is the cloning of suitable candidates in order to double research productivity. A recent publication by Moving Science, however, claims that the clones spontaneously mutate and undergo speciation events. Several clients of Moving Science came forward with claims that this report is fake news. We accessed archived records kept by Moving Science to uncover the truth.
Good morning, everyone. Thank you for joining today's research. Professor X, I don't think you're muted. So thank you. Dr. Smooth, I don't think you're muted either. Dr. Smooth? Good morning. All right, okay, let's continue. Today we'll be discussing Project 6449. I will now hand over to ARISTKA, who will be presenting their work. Please feel free at any time to interrupt and ask questions. All right, over to you, Dr. ARISTKA. Dr. ARISTKA, we can't hear you. I think you're muted.
Press the mute button in the left corner.
Is this okay?
Perfect, thank you.
Sorry about that. Let me share my screen with you quickly. Good morning, everybody. Let's share over there. Thank you for your participation in our research discussion this morning. Today, we are discussing the spontaneous mutation and divergence observed in Cloning Project 6449. As part of the Fees Must Fall innovation campaign in 2016, Moving Science performed a cloning experiment to test if human cloning was possible. The suitable candidate was called ARISTKA. Not only was the first human cloning possible, but the clone was successfully integrated into society. We did, however, notice that these clones were able to spontaneously mutate and they started diverging into two different species.
We decided to further investigate whether these clones are in fact undergoing a speciation event and could be considered as two distinct species. Our approach was to compare the following characteristics: their phenotypic differences, to observe their natural habitat, their recognition by other members of the population, their observed mating types in nature, their fungal associates, and of course, next generation sequencing to perform phylogenomic analysis.
So first, we examined the phenotypic differences in the two individuals. No phenotypic differences were observed once cloning was completed. However, we detected point mutations within the clones, leading to significant phenotypic changes after 7 days. And the ARISTKA and ARISTKA prototypes seem to have diverged even further in recent years. In spite of their phenotypic differences, the samples are still sharing several similarities.
We next investigated the natural habitat of both samples. Both were observed in the same types of environments, predominantly in laboratory conditions, but sometimes also observed in field and forest areas. We couldn't conclusively confirm the mating types of both samples. The one sample was confirmed to be heterosexual, but the other sample still needs to be confirmed in nature.
A strong indication of the differentiation of these two species was their fungal associates. While only Dothostroma and other pioneer pathogens could be isolated from ARISTKA, Ceratocystis as well as various symbiotic bacteria were collected from ARISTKA. Even though there are distinct differences between the two samples, the overlapping features seem to lead to incorrect recognition and identification of the two samples by other members of the population.
which again made us question the authenticity of the speciation of these two samples.
We therefore turned to a genetic approach to further distinguish between the two isolates.
Here we performed a next generation sequencing analysis to identify SNPs and constructed a phylogenomic tree. This conclusively proved that even though the two samples seem to be closely related, they can be considered as distinct species. And this also provided some clue to their ancestral species.
Hi, ARISTKA. This banner here. I was wondering, I don't know if this will pass for the peer review system. The sample looks quite small, but just a thought.
This is Dr. Strange. I was wondering, do you maybe perhaps have any other examples for us?
ARISTKA will share the slide. So this phenomenon has also been seen in the Quinton prototype. And recently we discovered that the Darryl prototype is confused with the unknown prototype of Darren. Additionally, Matthew and Alyssa prototypes seem to have diverging clones.
Additionally, we have 4 other prototypes that are more problematic and which results in more drastic speciation and divergence. We call them the Robert, the Nicole, the Juanita, and the Rachel. And that is all that we have for this morning.
Thank you.
Egg speaking. I really do think we should report these findings to the board. ARISTKA, could you please write it up?
Mm-hmm.
Sure,
Yes.
should be quick.
Everything is awesome. Everything is cool when you're part of a team. Everything is awesome when we live in our dream. Everything is better when we stick together. Side by side, you and I gonna win forever. Let's party. Everything's not awesome, but that doesn't mean that it's hopeless and bleak. Everything's not awesome, but in my heart I believe we can make things better if we stick together. If we stick together, side by side, you and I, we will build it together. Build it together. This song's gonna get stuck inside you. This song's gonna get stuck inside you. This song's gonna get stuck inside you. This song's gonna get stuck inside your heart. Everything's not awesome. Things can be awesome all the time. It's an unrealistic expectation, but that doesn't mean you shouldn't try to make everything awesome in a less idealistic kind of way.
Although our investigation confirmed that the publication by Moving Science is not fabricated, we still have doubts whether the clones truly speciated. We tested whether colleagues of ARISTKA and ARISTA that claim to closely work with the clones could successfully distinguish between the two. We showed them the image of ARISTKA shown here to the right and asked them to identify the clone.
Do you recognize her?
Is ARISTKA?
I don't know.
It looks like ARISTKA. I think.
I think I see ARISTKA here.
I definitely see ARISTKA. Oh goodness.
Lastly, we interviewed the clones individually, and the similarity in their response is unsettling.
Do you think you are unique individuals?
Yes, of course. I'm my own
my
person.
person.
As we conclude this episode of Fake News Uncovered, we leave you, as always, with the opportunity to decide for yourself. Keep on uncovering the truth. Good night.